Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, welcome to my presentation. I'm very grateful to you all for taking the time to be here. My name is Ika Yanti. I'm a researcher from Balai Besar Litbang Tanaman Obat dan Obat Tradisional Tawangmangu. Today, I'm going to present my study which titled In Vitro Anticancer Screening of Selected Indonesian Medicinal Plants. First of all, I'll talk about the background of the research. Based on WHO data, cancer is the second leading cause of death globally. Most common types of cancer in women are breast, colorectal, lung, cervical, and thyroid cancer. The development of proper cancer treatment management is still a challenge today. The discovery of variety of anti-cancer bioactive compounds from Indonesian plants is one of the potential strategies to be implemented. In 2012, 2015, and 2017, we have been conducted a community-based ethnomedicine study. It's called Riset Tumbuhan Obat and Jamu, or RISTOJA. It found that there are more than 400 species of medicinal plants used by the traditional healers to treat cancer. So, it's an abundant source to be explored for the anti-cancer activity. The aim of the research was to evaluate the cytotoxic potential of selected Indonesian medicinal plants. Now, let's move on to methods. The samples are 30 plant species. The selection of the plant was based on the availability in nearby. We collected 29 species from West, Central, and East Java, and only one species was collected from East Nusa Tenggara. The plants were dried, grinded, and then extracted by graded maceration using dichloromethan and methanol. So there are 60 samples of extract to be evaluated. The cytotoxic assay was carried out using MTT assay. We evaluated the extracts on 3 cancer cell lines: HeLa cervical cancer cell, WIDR colon cancer cell, and T47D breast cancer cell lines. The concentration was varied from 15 to 480 micrograms per milliliter. The cells were incubated for 24 hours. Cell viability was analyzed using linear regression to obtain IC50. The next point is results and discussion. Table 1 consists of plant species number 1 to 10 which evaluated against 3 types of cancer cells. It showed that there are 3 species that have IC50 value less than 100 microgram per milliliter in all 3 cancer cells. The plants are Calophyllum inophyllum, Tetracera scandens, and Calicarpa longifolia. Artocarpus altilis has IC50 value less than 100 microgram per milliliter in 2 cancer cells, WIDR and T47D. Meanwhile, Hyptis suaveolens has IC50 value less than 100 microgram per milliliter only in WIDR cancer cell. Table 2 showed the IC50 values of species number 11 to 20. There are some species that have strong cytotoxic activity. The highest activity was found in Leucas lavandulifolia, which has the IC50 value about 40 microgram per milliliter in all 3 cancer cells. In Table 3, we can see the IC50 values of species number 21 to 30. It showed that the strong cytotoxic activity was found in Gerardanthus macrorhizos and Micania micrantha. In WIDR cell, both extract, dichloromethan and methanol extract of Gerardanthus macrorhizos have IC50 value less than 100 microgram per milliliter. In other 2 cells, only dichloromethan extract has IC50 values less than 100 microgram per milliliter. Both extract of Micania micrantha showed strong cytotoxic activity in 3 cancer cell lines, except the methanol extract in WIDR cancer cell, which was inactive with the IC50 value more than 500 micrograms per milliliter. The previous study demonstrated that the butanolic extract of Micania micrantha leaf possessing dose-dependent anticancer activity attributed to the presence of polyphenolic groups. Besides that, the other study reported that aqueous extract obtained from Micania micrantha showed antitumor activity in vitro on K562 and HeLa cells and in vivo experiment. From the results, the plant extract can be categorized into three groups of cytotoxic activity. Group 1 is strong cytotoxic activity. This group consisted of extracts that strongly influence the morphology and viability of three types of cancer cells, both HeLa cervical cancer cells, WIDR colon cancer cells, and T470 breast cancer cells with IC50 value less than 100 microgram per milliliter. Extracts included in this group category are dichloromethan extracts consisting of 6 types of medicinal plants, namely Calophyllum inophyllum, Tetracera scandens, Calicarpa longifolia, Gerardanthus macrorhizus, Leucas lavandulifolia, and Micania micrantha. The second group is moderate cytotoxic activity. This group consisted of extracts having IC50 values in the range of 100 to 300 microgram per milliliter. This activity was shown against at least 2 types of cancer cells. This group includes dichloromethan extracts and methanol extracts. The plant species are Hyptis suaveolens, Disidia rucifolia, Moringa oleifera, Lausonia inermis, Malvastrum coromandelica, Crescentia cujete, Jatropha curcas, Meremia peltata, Morus alba, and Cyanthillium cinereum. And then the last group is weak cytotoxic activity. Extracts belonging to the category of weak potential have IC50 values in the range of 300 to 400 microgram per milliliter. Extracts included in this category are Phyllanthus acidus, Duriozibetinus, Inocarpus fagifer, Chromolina odorata, Asplenium nidus, and Rhizopora mucronata. Now, I'd like to finish with the conclusion of this study. 6 plant species have strong cytotoxic activity against 3 types of cancer cell lines. Further research is needed in identification of active compounds and their anti-cancer mechanism. That's the end of my presentation. Thank you very much for the attention. Wassalamualaikum warahmatullahi wabarakatuh.